Hey guys, I did my project on the Impossible Burger, which is a product that was founded by the company Impossible Foods. The production of the Impossible Burger we know today was driven by the discovery of the lake hemoglobin in the roots of soybean plants. You may recognize the heme or hemoglobin rooted in that word. Much like animals who have hemoglobin and myoglobin in their bodies, soybean plants have this property as well in the form of lake hemoglobin. The heme molecule is what they share. This allowed scientists to further take advantage of the similar property to create something never imagined before, an Impossible Burger. It was created by Impossible Foods, founded by Patrick Brown. With the ability to mimic the beef that people love, there was an incentive to put forth the creation as a force against the beef industry, which contributes many issues for the health of people that consume it and that of the earth in general. The product has the perceived capability of putting a stop to domestic animal cruelty and reduce the overall carbon footprint. Assuming all of us have tried a burger before, we know the classic flavor of it and the texture. If you haven't tried it, you must at least know the smell of them cooking on the grill. These are the senses that make a burger to us. As the beef cooks on the grill, there is a protein interaction that creates the harmony of flavors it is famous for. What is believed to bring its unique flavor is the heme molecule. In animals, this molecule can be found in hemoglobin and myoglobin, which hemoglobin is found in the blood and myoglobin in the muscles of our body. To go into more depth, let's look at the similarities of the molecules hemoglobin, myoglobin, and lake hemoglobin. New scientific advancements have given scientists reevaluation of the structure and function of them all, changing our understanding. Because both molecules, lake hemoglobin and myoglobin, are rooted with heme, we understand that they can serve similar functions. Lake hemoglobin structurally is most similar to myoglobin. Lake hemoglobin protein consists of a heme bound to an iron, and one polypeptide chain, the globin. Similar to them both, myoglobin and hemoglobin, the iron of heme is found in its ferrous state in vivo and is the moiety that binds oxygen. To relate back to the topic of recombinant DNA in Chapter 9 of our class, we talked a lot about DNA cloning. Recombinant DNA is artificially created DNA sequences that do not normally occur together in nature. Seen in the illustration here, there are the steps we are familiar with creating a recombinant vector. Impossible Foods has utilized a similar approach in order to multiply the amount of lake hemoglobin they need to create the Impossible Burger, which is a transgenic food. In the next few slides, I will discuss how the lake hemoglobin is incorporated by Impossible Foods using a yeast vector and a fermentation process. In order to obtain the gene of interest, in this case the gene for lake hemoglobin, the use of a restriction enzyme or restriction enzymes is necessary. These are enzymes that act like scissors and cut around the desired gene to isolate it. For this process, they use familiar restriction enzymes that we have worked with in lab. These are EcoRI and HIND3. Restriction enzymes can either have sticky ends or blunt ends, both of which in this case result in sticky ends. They have what is called an overhang, which is a short region of nucleotides that are left unpaired. These sticky ends are what allow the recombination to occur with the yeast vector. Pichia pastoris, specifically MXY0291, is the yeast vector that Impossible Foods uses for fermentation that results in an accumulation of lake hemoglobin. It is a common vector used in genetic engineering as it is easy to manipulate. The fate of the Pichia pastoris vector is to undergo transformation of the desired DNA, and then the process of fermentation to multiply the recombinant vector which contains the target DNA. In order to prepare a Pichia pastoris strain for transformation, a plated media is inoculated with a specific strain. It is grown overnight in a shaking incubator. After the growth, the cells are diluted into a liquid form of the same media and grown again in the same temperature conditions, but only for about 4 to 6 hours. The cells are then centrifuged at 500,000 RPM for about 5 minutes at room temperature. The cells are resuspended in solution and centrifuged again, and this is repeated twice. Once completed, the cells are considered competent, meaning they are ready for transformation. For each transformation, the following protocol is followed for Pichia pastoris. First, 50 microliters of the competent cells that were just produced are put into a microcentrifuge tube. Next, 3 micrograms of the desired DNA is incorporated into that mixture. Linearized DNA can be used directly from the restriction digest that was performed without affecting the transformation efficiency. 1 milliliter of solution is added to the mix and vortexed. The mixture is incubated for 1 hour at 30 degrees Celsius. Every 15 minutes, the mixture should be vortexed to ensure efficiency of the transformation process. Once time is up, the tube is heat shocked in a 42-degree water bath for 10 minutes. The cells are pelleted by centrifugation at 300,000 RPM for 5 minutes at room temperature. The supernatant is discarded, and 100 to 150 microliters of the solution resuspends the pellet. Using a sterile spreader, the entire transformation is plated on selected plates and incubated for 2 to 4 days at 30 degrees Celsius. What is yielded is approximately 50 colonies of the recombinant vector. The next step in growing the desired DNA is by fermenting the recombinant yeast that was produced in the transformation process. An O2 saturated medium is required to begin the process. The Pichia pastoris will consume oxygen as it grows, reducing the dissolved oxygen content. However, because oxygen is required for the first step in methanol catabolism, it is important to maintain the dissolved oxygen level or concentration at a certain level to ensure growth of Pichia on methanol. The medium is in a flask where it is inoculated with a frozen glycerol stalk. The flasks are grown at 30 degrees Celsius. The content of the flask is then transferred to a fermenter, which undergoes the same fermentation process at a much larger scale. DO levels must stay at 20% because the yeast will use the oxygen. The yeast is in the fermenter until the glycerol is completely consumed, which can take about 18 to 24 hours. Though this cycle is complete, the recombinant protein will not yet be produced due to the absence of methanol. After the batch growth phase, which we just went over, there is an initiation of glycerol fed to the fermenter that will increase the biomass of the cells. This is important because the level of expressed protein is dependent on the biomass of the cells. The DO must remain stagnant during this phase as well. This can take about 4 hours. The cellular yield of 180 to 220 grams per liter wet cells should be achieved at the end of this stage. Once all the glycerol is used up, methanol is slowly incorporated, as if to wean from glycerol to the methanol. The methanol feed is then slowly increased over time after being introduced. The entire methanol-fed batch phase takes about 70 hours of time, with a total of approximately 740 methanol fed per liter of initial volume. Because the cell density increases during this time, the end product should be about double of what it started with. Produces the like hemoglobin in a large abundance. Like hemoglobin can be purified from the soybean root nodules after these steps that were taken. Purified lake hemoglobin is fully oxidized by using an excess of potassium ferricyanide. Finally, this oxidizer is removed from the solution and the lake hemoglobin is considered purified. Lake hemoglobin is ready to be added with the other essential ingredients in the Impossible Burger after this stage. The purified lake hemoglobin is added to make the Impossible Burger. Some of the other ingredients that are used to make the patty are soy, water, sunflower oil, cellulose, potato protein, etc. Of course, the lake hemoglobin is added as well. In creating the patty, it looks and tastes like blood as it is added to the mixture, giving it the red and meaty characteristics. Once the mixture for the meat is made, it is churned and mixed just like the hamburger meat would be. They are pattied, frozen, and ready to be consumed. The motivation behind the Impossible Burger was to make a statement that something as desirable in the world, such as beef, could be entirely mimicked by plants. Pat Brown, the creator, explains that the Impossible Burger was made to address the greatest environmental threat that humans have ever faced, which is the catastrophic impact of the use of animals as a food technology on the global environment, on climate change, on water resources, land use, and biodiversity. Cows are the most destructive species and the most invasive species on Earth, except for possibly humans. Brown's hope is that over time the industry will be replaced by one with better technology, which he explains is a true trend throughout history. In the future, the production of the Impossible Burger could hypothetically replace that of the beef industry. In conclusion, the science behind the Impossible Burger is one that can compete with the beef industry. By the use of genetically engineered yeast and fermentation, lake hemoglobin can be produced in fantastic amounts and can be contributed to a soy-based burger to mimic a real beef patty created by the beef industry. By the discovery of lake hemoglobin in soybean plants, our knowledge and understanding of heme has been expanded, and the discovery that a plant can produce something that an animal can is revolutionary. In theory, the global carbon footprint could be benefited by this plant-based industry by competing with the beef industry. The use of cows for our consumption is a big contributor to the Earth's biggest environmental threat and can be lessened by the discovery of this plant-based Impossible Burger and other revolutionary products on the forefront. The subfields of biochemistry that this topic belongs to are plant biochemistry, genetics, as well as molecular biology and metabolism. And there are my resources.